All right, I think we'll just get started. But thanks everyone for coming. I'm Jason. I'm one of the new co-organizers for this seminar series. And today I'm really excited to welcome Andre Cornman, who is one of the co-founders of Tatta Bio, and he's gonna talk about the OMG dataset and a little bit about the genomic language models that they're developing there. So Andre, you please take it away and feel free to introduce yourself a little bit more as well.
Awesome, thanks a lot. Yeah, hi everyone. I'm Andre. As Jason mentioned, I'm co-founder at Tatta Bio. So we're a scientific nonprofit working on building AI tools and data sets to kind of accelerate biotech and biotech R&D. And today we're going to be talking about the OMG dataset. It's a dataset that we recently released, as well as GLM2, which is a model that we trained on this dataset. So first, I want to just go over kind of some background. So what are metagenomes and what is metagenomics? So metagenomes are sequenced directly from environments without requiring cultivation. So this allows for a huge amount of diversity compared to having to cultivate a microbe that you might know how to cultivate, which is like something like E. coli. And because we have this incredible amount of diversity, we can, if you draw a comparison to NLP, UniProt is something maybe like Wikipedia that's like highly structured, labeled, well curated, whereas metagenomic data is more like Common Crawl. It's like extremely unstructured, unlabeled. It does have a lot of diversity, but it also does have a lot of bias or potentially like poor quality data. So that's kind of the promise of metagenomics. So what does the data actually look like? So we start with a sample from some environment, for example, like the bottom of the seafloor or a human gut. And you get in that sample many different organisms that live in that environment. This goes through assembly to basically take these DNA reads and combine them into longer sequences. And for a specific genomic sequence, you can then call a gene caller, which picks out the genes within that genomic sequence. And this is in both the forward and the reverse direction in the DNA sequence. And then you can kind of represent this sequence with genes as a multimodal sequence. So in this case, we have 3 genes going in the forward direction, 1 gene going in the reverse direction, and then these intergenic sequences. And so in this talk, we'll be representing, or I'll be using CDS as a coding sequence, which represents the gene, or IGS, which is an intergenic sequence, which is a non-coding DNA. So why should we be interested in pre-training on metagenomic data sets? So first, there's just a huge amount of diversity, and we know that large language models, like they need these huge data sets to kind of learn strong representations of the data. So this is really exciting, but the main cost is that we do need to do very careful balancing and deduplication because it's so unstructured. The second kind of exciting point is multi-gene context. So specifically for microbial genomes, context is extremely informative of function and protein-protein interactions. So when you train on protein-only data sets like UniRef, you kind of throw away all this data that where we do know that proteins that occur in the same context co-evolve and co-occur and interact with each other. So this is kind of information that can be really exciting to learn. And finally, we can also train on intergenic sequences, which are the sequences that do not code for proteins. And these can contain interesting regulatory and evolutionary signals that can be learned by the model. So in this talk, just a quick overview, I'll be first talking about the OMG dataset. Then I'll go over the GLM2 model, which is a mixed modality language model. And I'll be talking about some results in unsupervised protein-protein interaction. And finally, some exciting upcoming work. So to introduce the OMG dataset. So it's the largest metagenomic dataset to date. And what we did is we combined the two largest repositories, JGI's IMG and EMBL's Magnify. So after combining these datasets, we represent it as a mixed modality dataset for multi-gene modeling. So we take these datasets and we, as I mentioned before, we represent the coding sequences as amino acid sequences and then non-coding sequences as nucleotides. And these basically occur in a sequence. We do a lot of quality filtering to remove poor assembly and poor quality data. And we also make it easily accessible on the Hugging Face hub. So kind of to visualize what this data set looks like, so we actually released two versions of the data set, one called OG and one called OMG. So the OG data set is a subset of OMG that has taxonomic labels. So these are from so for every contig we have its taxonomy. So we can plot this with a UMAP on the left using a model that we trained on this data. And so first, one thing to notice is that we do see nice clustering by taxonomic class, which is nice to see. But then on the right, what we do is we plot the same OG dataset and overlay it in gray with the OMG dataset. So basically comparing metagenomic data to non-metagenomic data, and we see a lot more diversity at least in the UMAP plot. So this kind of shows that we have a lot of new data in the OMG that might not be represented by kind of more well-curated data sets. I'm just looking over some key statistics of the OMG dataset. So it contains 3.3 billion proteins, 2.8 billion intergenic sequences, and this covers around 3.1 trillion base pairs. We also cluster this OMG dataset specifically with only considering the proteins, and we cluster it at 50% sequence identity. And this ends up with 207 million protein clusters, which is about 3 times larger than UniRef 50. So what this really shows is that there is a lot more diversity in sequence space in the OMG dataset compared to something like UniRef. So yeah, I'll move on to talking about GLM2. So this is our model that we trained on the OMG dataset. So GLM2 is a mixed modality genomic language model. And specifically, it has multi-protein context. So instead of just encoding a single gene, we actually encode a whole genomic context, which includes multiple genes, both in the forward and reverse direction. So you can kind of see on the right, this is the information flow through the model. So we start with a metagenomic contig. Then the genes get called. So this is kind of where the OMG data set starts. So we have a sequence of coding sequences and non-coding sequences where the coding sequences have an associated direction. We tokenize this into amino acids for the coding sequences and DNA and base pairs for the non-coding sequences. And then we randomly mask like 30% of the tokens. And we ask the GLM2 model to reconstruct the masked tokens. So you can think of this as quite similar to like protein language models, except now we really have like both multiple genes and as well as intergenic sequences. And we trained on the OMG dataset after semantic deduplication, and I'll talk about that in the next slide. And we also release 2 scales in the model, 150 and 650 million parameters. Just to briefly go over semantic deduplication. So one of the key challenges I mentioned about metagenomic datasets is that they're really hard to dereplicate. So the problem is really that you have these arbitrary length and like regions of sequences that you need to deduplicate, and that's really computationally challenging. So unlike protein data sets where you can just simply cluster the proteins, that approach isn't really feasible. So we decided to take a more like a different approach here. where we first train a model on all the data set, and then we embed the whole model, embed the whole data set, and basically prune points that are within epsilon distance of another point. So visually, we can see what that looks like. So on the left, we perform this on the OG data set, so we can see what kind of taxonomic labels get pruned. So on the left is the same OG data set with labels, and on the right, we highlight the prune points in blue. So as we expect, what we see is we see a lot of a high density of prune points in these overrepresented taxa. So that's kind of promising, and it does what we expect it to do, which is kind of do more balancing over the taxonomies. And if we actually look at the distribution of taxonomies, we do see that both for phylum distribution and order distribution, we do see a good amount of balancing. So one thing to note is like first, like using taxonomic labels for OMG is not really possible for balancing because we don't have taxonomic labels. And the second is balancing with taxonomic labels can also be challenging because it's unclear how you want to balance these, whether it should be like even distribution or not, because some taxa have way more sequence diversity than others. And to evaluate the model, we evaluated on the DGB dataset, which is a benchmark that we released earlier this year. And the DGB dataset is basically designed to capture function across as many tasks as possible and across as many diverse sequences as possible. So we try not to bias towards model organisms like E. coli. So some of our tasks are, for example, like EC classification or evolutionary distance or like operon prediction. And so when we look at the results, so the first thing we note is that GLM2 does score higher on the aggregate DGEB score. So the points on the left are the smaller GLM2 model. And what we see secondly is that semantic deduplication does improve the overall score. And when we look at which scores improve, we do see a consistent improvement for underrepresented taxa, which is kind of what we'd expect given what we saw from the pruning. And let me just maybe answer one question in the chat. Daniel asked, what is the reason for converting coding sequences to amino acids as opposed to leaving everything as nucleotide? Yeah, that's a great question. So this is, I guess, yeah, so that's exactly right. You could, in theory, just train on raw DNA, and I think that's what a lot of models have done before, or some models have done before, like Evo, for example. I think maybe the reason we've done this, like, there's a couple reasons. From just an engineering standpoint, like, you end up with much longer context lengths if you train on raw DNA. So that's much less efficient to train. And then secondly, what we've seen is actually these DNA models, like internally, they just like during the initial stage of training, what they do is they find the models learn like which parts of the DNA represent coding sequences and which parts do not. And we're basically just feeding the model directly that information so it can like doesn't need to learn that. There always are issues with like gene calling, but we thought this was like a reasonable trade-off. And then Lucas asks, what was thinking for sequence tokenization? Would K-mers make the vocabulary too large? Yes, that's a great question. So there's definitely different ways you could tokenize sequences. I think it's not necessarily a problem with vocabulary size because our vocab size is quite small compared to like GPT, for example. So one thing we can note is like tokenizing per base pair per amino acid just makes things simpler, especially if you're looking at like coevolution between different amino acids or different base pairs. And this is actually related to the next part of this talk, the protein-protein interaction, where the model needs to reason about how different positions interact with each other. So that might be abstracted away if you use k-mers tokens. So let me just continue on. So I'll move on to the PPI prediction. So once we train our model and we benchmarked it, we want to take a step further and see whether our model can learn protein-protein interaction without supervision. So we already know that protein language models trained on like protein-only data sets do learn coevolutionary signal, which can be extracted by this method called categorical Jacobian that was released recently from MIT. And we also note that existing tools for protein-protein interaction are limited. So often they require like supervision, but the known PPIs are extremely sparse and biased. So we don't really see these working well for kind of underrepresented taxa. And so the key here is, can we do this in an unsupervised way? So we first looked at this mod AC complex, which is a transporter complex, and these two proteins interact and co-evolve at these locations where they're physically in contact. So we wanted to see whether GLM2 can pick up on this. So here we first look at the POTS model, which is an MSA-based approach. And the MSA approach does pick up this signal that between the two proteins where we expect the coevolutionary signal to appear. And we also see that GLM2 does excitingly pick up this same signal. Whereas if you look at ESM, this signal is not there, which is not really surprising because ESM is not trained on multiple proteins in the context. Also, EVO doesn't pick up on the signal as well, and they've trained this with DNA only. So maybe this is another reason why it could be more efficient to train on translated proteins instead of raw DNA. So we also want to see whether our model can pick up structural information about intergenic sequences instead of just looking at protein sequences. So in this case, we're looking at a stretch of E. coli, and we see that the model does pick up on tRNA structure as well as the terminator and transcription binding sites. So that's kind of exciting that we can identify these in zero shot without any supervision. And yeah, I'll go over share some upcoming work that we're extending our GLM2 model. So once we released this, we found that the Jacobian is a really exciting way to kind of interact with the model, but we wondered what other ways can we interact with GLM2. And one way is, can we use GLM2 to search across sequence, structure, and context? So we're building this search tool called Gaia to search for proteins that have a similar sequence structure context, and using GLM2 embeddings as a vector search, and hopefully be able to use the context to identify possible functions of an unannotated gene. So just to kind of quickly show what it looks like, given a search query, we can look at the protein structure as well as like 100 or so retrieved genomic sequences and contexts. And then we can kind of look at the context to hypothesize about a certain gene's function. So this is kind of extending the way we can interact with GLM2 to hopefully work on this problem of annotating sequences that are unknown or have very limited information. So yeah, I'd like to just finish by acknowledging like all our awesome collaborators. So this was work from Tatta Bio as well as JGI, EMBL, Seoul National University, and MIT. Yeah, thank you. So yeah, I guess I can, if there's anyone who has questions, we can go over those, or I can also maybe go through the chat.